Okay, so that's all for recombinant DNA, but there's another process we need to break down involving DNA before we move on to our more uses of this recombinant DNA, and that's DNA sequencing and profiling. DNA sequencing is a pretty nifty way to figure out the exact order of A, G, T, and C in a chunk of DNA. So we're really looking at a very low molecular level that even our microscopes wouldn't be able to see. Scientists use these really fancy fluorescent dyes to label each of the building blocks with different colors. They then run this DNA through a gel in a process called gel electrophoresis, which we'll dive into later. And as that DNA moves through the gel, it scans a laser and picks up the colors, which tell us the sequence of those bases. Why does this matter? Well, DNA sequencing isn't just for catching criminals, although it's great for that too. It's used for everyday stuff like identifying people for things like ancestry tests or figuring out the family tree of plants and animals. In forensics, it's crucial for pinpointing who left their DNA at a crime scene, which helps solve cases. Essentially, if you have a strand of DNA and you find the sequence of bases, you're able to identify how closely linked or related two individuals are, because obviously there'll be more commonality between two individuals that have a certain sequence of DNA or based on their parents, as we would have learnt with inheritance. Okay. DNA sequencing also helps scientists to explore biodiversity, like discovering new species or tracking endangered ones. Plus, it's a game changer for understanding how animals reproduce. Like I mentioned, who's the biological parent and who's involved in raising the offspring. There are a lot of interesting applications for this technology, but in order to use it, we do need to understand gel electrophoresis and PCR. So